This is a very informal get together. It's supposed to be a fireside chat. I've never been at a fireside with so many people, but we'll make this work.
Need a bigger fire.
And please feel free to remind us if you can't hear. We'll do our best. Okay, how are we doing to get this started? We're started already. Okay, great. So we've had a very busy day today, and I told Dr. Venter I wouldn't work him too hard, but
he lied.
But we met with Susie Walker earlier, and I said I think I'm more tired than he is. So, anyways, it's really a great honor and pleasure to have Dr. Craig Venter here today visiting us. For those of you who don't know, Craig, he's actually on our Science and Innovation Advisory Committee here at SickKids. And he's here in Toronto because yesterday he was awarded the Edagawa Prize, and he received that yesterday here. So he agreed to stay on for the day to spend time. We spoke to the board of directors this morning. We had an event at the Arts and Letters Club to announce.
You're all getting a pay raise.
His new book, and he agreed to have one of our discovery dialogue sessions with us today. And I've done many of our previous famous scientists who have been here at SickKids. This is one of the first times that we've had someone from the outside, although we've collaborated together on some scientific papers before. So I'm not going to spend a lot of time introducing Dr. Venter, but just to remind you that he's actually a biochemist by training. We'll talk a little bit about this. And he has become a geneticist or genome scientist when he was at the Roswell Park Institute and through the NIH. And of course, he was the leader of the Scherer group that sequenced the first genome, first human genome. Prior to that, he sequenced the first living organism, Haemophilus influenza. He developed the express sequence tag or EST sequencing concept. After, or kind of congruent, he sequenced the mouse genome, rat, many other organisms. And see if I get the order right, he then launched off to kind of circumnavigate the globe and sequence microbes. And we're going to talk about that's in his new book a little bit later. Synthesized the first free living organism, and the list goes on and on and on. So we're really, really fortunate here. We decided to have this event actually on the 13th floor because this is the Genome Center at SickKids. And Craig actually earlier came to SickKids when we first got one of the sequencing machines. I was one of the ABI machines many years ago. I think it was 2002 or so. But welcome back.
Thank you.
So
this building wasn't here then.
No. And we'll be going to the Gardner Awards dinner tonight. So, of course, he's a recipient of the Gardner Award 2002, if I remember correctly. Okay, so Craig, we're going to just break this up. I'd like to ask you some questions around the concept of discovery first. And then we'll, near the end, we'll talk more about your book, if you will. So I went back and reread one of your other books, the story of your genome. And in chapter 6, the chapter is called Big Biology. This is in your autobiography. It's called The Life Decoded, My Genome, My Life. And I thought that one of the quotes that led into that chapter was quite relevant to discuss in the concept of discovery dialogues. And it's actually from Sidney Brenner. And it says, progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. Do you want to comment on that?
Well, techniques are the new tools that allow us to ask questions we couldn't do before. So, when Darwin was in the Galapagos way before me, he was limited to his visual acuity for what he could see. We had the new lens of DNA sequencing, so we could see this invisible world that nobody else could see. But there's case after case. The recent new telescope that goes back 8 billion years and the history of the universe and all these amazing pictures of galaxies that weren't possible. So the new tools constantly have allowed us to see further. Uh, you know, um, one of the nicest awards I got was the Leeuwenhoek Award. It's only given once every 4 years, and I think I'm the only living recipient right now. So, but they give you a copy, a hand-tooled version of his microscope, which is only about this big. But it still has a little cell in it that you can see from the slides that he made. So in molecular biology, the tools are a microscope,
Right.
and what would grow on culture for the last few centuries. And it's only, the microscope allowed to see things that nobody could see before. But DNA sequencing allowed us to see things that weren't even visible on the microscope. So tools and advancement and using those for trying to answer new questions, I think are the probably number one thing that advances science.
Yeah. I think we'll come back to this in the discussion later also. Actually, in the same chapter, there's another great quote. I didn't just read that chapter of the book, but these one really actually hit me quite.
I don't even remember these.
Yeah. So, it goes like this. It says, in science, the credit goes to the man, I'll say person, who convinces the world, not to the man to first come up with the idea, not the man who finds a grain of new or precious quality, but to him who sows it, reaps it, grinds it, and feeds the world on it. I think this is kind of.
Who was that from?
So this is from Sir Francis Darwin, the third son of Charles Darwin.
Yeah.
It's actually delivered at the first Galton lecture in the Eugenics Society back in 1914.
Yeah.
And it's really kind of interesting. And I actually teach in one of my first year courses that I teach here. I use my own phrase that a discovery is not simply a eureka moment, but instead a process where you capture a glimpse of nature exposed, convince your peers of what you saw, and demonstrate utility. Eureka on its own is merely an observation. So how do you frame, or how do you think of discovery?
So I had a great scientific mentor, the late Nate Kaplan, who discovered coenzyme A. And he had over 100 people in his lab, but he spent a lot of time with me. And he probably had 2 or 3 great new ideas every day. I don't know if I have that many, but, with the various things that I've done, I've had people come up after the fact and claim, well, they thought of it before I did it. And what counts in science, as the quote says, it's not thinking about it. It's not having the idea. It's putting ideas into action. Right. And so there's sort of bottlenecks of thinking or parallel thinking where a lot of people will have similar ideas. Sort of the advancement of ideas gets, people have very similar conclusions. But the ones that make the breakthroughs are the ones that actually put those ideas into action. And the other notion that Kaplan taught me is that most scientists talk themselves out of doing the experiment because some people are afraid to succeed, some are afraid to fail, some just, various reasons talk themselves out of it. But it's the same thing. If you have the idea and then you talk yourself out of doing the experiment,
right,
you won't become part of history.
Yeah.
So if you have an idea, even if other people have similar ideas, the first to act on it is the one that's going to make the changes.
Yeah, it's really nicely put. And so what you're telling
George Church said he thought of the genome when he was 8 years old.
didn't act on it.
He didn't act on it.
Yeah. So what you're telling that we have many trainees here is to do the experiment. And what I learned from my mentor, Lapchi Choi, was look at the data too. Mm-hmm. Don't just do the experiment and wait. You got to look at the data as soon as you can.
Yeah.
Okay. So in your opinion, what was your most significant discovery or discoveries? And then I'll give you my interpretation.
Wow. Well, it's much broader than a single scientific discovery. I think it's learning to follow my intuition and follow through with my ideas and do something with them. I mean, it's I don't know which of the things I've done that I'll be most known for. Probably the most original and most significant was making the first synthetic cell.
Mhm.
And that came from an idea after sequencing the Haemophilus genome, and we did the mycoplasma genome the same year, thinking that comparative genomics would give us answers to interpreting the genome. And it just made it worse. The more genomes we sequenced, the worse the problem got, because every genome had a unique set of unknown genes. And so it was at that stage back in 95, following the quote that we wrote into the synthetic genome from Richard Feynman, to understand it, you have to build it.
Right.
And so that was probably a single decision that probably had the biggest impact when I decided we have to try and synthesize a genome to be able to understand it, because you have to know all the components to do that. And that's also why it took so long. And as people know, even with the final genome, about a quarter of the genes essential for life were of unknown function.
Mhm.
So we couldn't, we learned that we didn't have sufficient knowledge in the field to a priori design a living cell. But I think the decision to do that early on was probably ended up being one of the most profound in my own view.
I would agree. I think the conceptualization and utilization and implementation of the whole genome sequencing shotgun approach coming from the top down was probably your most impactful,
Yeah,
in my opinion.
thus far, yeah.
The EST, so we were reminiscing earlier at lunch. In the early days of the Genome Project, I was very privileged to go to some of the early meetings. The approaches people were talking about were just based on mapping, really, and it was Craig Venter's kind of conceptualization of doing express sequence tag sequencing, which really started to change everything in the early days. And it was sequence based, but it was still based on assembling data using computation. So you married two different technologies here, and I think that was also a game changer. Do you want to comment on that?
So mathematicians love my work, even though I'm not a mathematician. I'm probably the opposite of one, because everything's based on randomness, which is a key concept in mathematics and physics. So ESTs were just randomly selecting clones from the cDNA library and sequencing, not knowing anything else about them. This was after spending 10 years to try and get 1 out of 1,000,000 clones for the adrenaline receptor to try and sequence it. And, gee, all these other things are just on there. What if we just randomly select them? Shotgun sequencing is based on having libraries with a perfect Poisson distribution, something Ham Smith was the only one that could create. But then you could randomly select those clones and then reconstitute them mathematically in the computer. So randomness is a key part of things. The ocean survey was just taking random samples in the ocean and sequencing to see what's there. So I guess you could describe my career as random.
Okay.
But there was a book early on that's probably nobody here has read it. Kaplan trained with Fritz Lipman, who discovered the high energy phosphate bond in ATP. And he had trained with Meyerhoff, but Lipman wrote a book about the strange route of his career. And it's called Wanderings of a Biochemist.
Right.
And just how one thing he did in science led to the next thing that led to the next thing. And I'd say my career sort of followed that path. It may seem incongruous to be sampling the ocean versus sequencing the human genome versus making synthetic life, but they're all around the same thing of trying to understand life at its most fundamental level.
So in your opinion, what was the purest discovery of all time when, the day before changed the day after when the discovery came out?
Well,
I haven't given any time to think about these things.
Yeah.
So
Probably the thing that there's two things, two areas. I think the thing that affected probably most people's thinking, that before we did the first analysis that Steve took part in, the first human genome, people were estimating that there were hundreds of thousands of human genes. Thinking there was going to be one for each human trait and characteristic. I'd say probably the biggest surprise to most people was there was such a finite number of genes, and that we weren't much different from fruit flies or rats. That's probably changed thinking more than anything else. And I've said this earlier in a conversation we were having, that what people consider major breakthroughs are actually just overcoming the bullshit ideas of the past.
Right.
Why people thought there would be 300,000 genes, or there'd be no life below 18,000 feet in the ocean. I mean, the trouble is science, particularly early in the history, was based so much on personalities, and the leaders in science, like Pavlov, they established ideas and dogma that people were afraid to challenge or couldn't challenge without data.
Right.
But they just established bizarre thinking. Like after we published the first genome, people labeled 98% of the genome as junk DNA.
Right.
I mean, these are most embarrassing ideas because they're based on proving how ignorant we are. Because we don't know what it is, we call it junk. And I said earlier, some of these people I offered to surgically remove their junk and see how long they lived, but I didn't get any takers. I think the other thing was the proof from the very first experiment in the oceans, showing the vast diversity of life, and that we were clearly a microbial planet. And once again, that the ignorance about life, that it could be off by so many orders of magnitude, is pretty stunning.
Right.
And somebody asked me the question last night of, from all these kind of discoveries, proving how much we didn't know. He goes, what else is out there that we don't know? Right. And I said, how am I supposed to answer that question? You have to devise new experiments to ask, right?
Yeah, Cindy Brenner had a great come back to the garbage or junk DNA. That garbage we throw away, junk we save.
That's right.
It's
junk
there
you keep in your attic.
Yes. I'm curious, what's your favorite scientific paper of all time that you go back and read of someone else's work?
Well, not a paper, but a book. I'd say Schrodinger's "What Is Life" is one of the truly most profound. And he had some ideas, right, suggesting that the genetic code could be a very simple code. He likened it to the Morse code. And I was talking to Francis Crick about this, and he goes, well, everybody thought that, but everybody didn't think that. And it's fun because I was asked to give the second Schrodinger lecture. He gave the first one in the same auditorium where he gave it, and still trying to define life, and the minimal cell and things help do that. But it's still a long way to go before we don't have an elevator definition of what life is. Yeah. It's a complex discussion and equation versus a simple short answer. We know what it's not. We know what death is. It's the absence of life.
Right. I think if for those of you who have not read the book, you'll hear all the great scientists that this is a book that's influenced their philosophies and their life.
What is life?
What is life? Yes, you must read it.
It's a very short book.
Yeah, you can read it in a weekend.
Read it very quickly.
I'm curious, you started out as a biochemist, and how do you see yourself now? Are you how do you define yourself, as a geneticist, a genome scientist, biochemist, or just a scientist?
So my PhD was in physiology and pharmacology. So I'm definitely not a geneticist. Some of the nicest awards, I've gotten some of the best microbiology awards. I've never taken microbiology. I learned microbiology one genome at a time. I don't like the term geneticist, so sorry if I'm offending geneticists, but I hated genetics. It was one of the most boring classes I ever took. It's probably the way it was taught, but primarily because so much was unknown still. So I describe myself primarily as a genome scientist, probably. But my first interaction with Jim Watson, he wanted to see the data. I was the only one that could get the early automated DNA sequencer to work. And he said he'd just come from Lee Hood's lab. Lee Hood wasn't even using his own DNA sequencer. He was still using manual methods. And he wanted me to explain how I got it to work. And I explained to him that I was not afraid to use a spectrophotometer, and that I just assumed that stoichiometry was important. And but people who did straight molecular biology would never do anything precisely. They would estimate the amount of DNA from the depth of a band on a gel. And even with Ham Smith, the Nobel laureate, I tried it. They couldn't get within an order of magnitude of what was actually there. And the first primers that came with the ABI system, turns out, had a secondary binding site. Mm-hmm. And so if you didn't have the stoichiometry precise, you just got gibberish. Mm-hmm. Mm-hmm. And it's only because I assumed that you had to have it right. I didn't know about the secondary binding site. It just, was applying rigor to the experiment, and we got beautiful data. And people more or less assumed it was some magic trick that I was using. But anyway, it was the first time, not the last time Watson insulted me, but it took me a while to realize that it was an insult. He goes, well, that explains it. You're a biochemist. So molecular biologists hated biochemists and vice versa. In part, it's understandable because biochemists set back the discovery of the genetic material for, maybe a century, a half a century, by insisting that DNA was too simple of a molecule, and proteins being much more complex, that proteins had to be the genetic material. Even though Drosophila researchers kind of knew early on and were dealing with chromosomes, that kind of thinking, again, fixed ideas, set back science for a very long time. But he's called me much worse things since than a biochemist.
And I think it's in one of your books. He came around when you mentioned who you did your PhD with, and he said, I respect you now. So we're in at the Hospital for Sick Children, if I forgot to say that, and we do a lot of sequencing here, and everything is based on having consents and protocols and things. And in your early days at Celera, your genome was one of the genomes that was selected. I think there were 4 or so, but yours.
Well, I was doing the selecting. So.
Yeah, you had to get a good one. But tell us a little bit about that. And I remember you did have a consent, and I think your mother was involved and things like this.
So it's hard to get in the mindset of people at that time, but you might recall that people were very afraid of genomics.
Yeah.
And that its primary thing was going to be misused. It was almost a century of Cold Spring Harbor being the center of eugenics in the U.S. and the genetic code or genetic traits being used against people, and so there was a big debate of how do you get somebody to volunteer to have their genome sequenced when there was all this fear around it. So the first genome, the one we did at Scherer, was actually 5 people. It was 2 Caucasian males, Ham Smith and myself, and 3 females, a Chinese woman, an African American, and a Caucasian. They have not self-identified, and I don't know who they are, but they chose not to self-identify. But it was a complex mixture, and it was sort of like there's a long history of self-experimentation in science. The case people probably most aware of was H. pylori, but Ham and I discussed it that it was difficult to ask other people why they should donate their genome if we weren't not willing to step up and do it. So it wasn't done out of egos, or we're both curious, and we're curious to see our genetic code, but it's because, if you're not willing to do it, how can you ask somebody else, to volunteer, and why should they do that? But, but it was complex because we set up a broad review committee, and, how do you get consent for something that's never happened before in history? Right. And, the worry is that it would affect future generations. And so it's, now people don't think twice about it.
So it never came back to bite you in any way? Insurance claims?
No, but there's been hundreds of papers published on all the diseases I should have died from. No, it's try to set a standard by showing, not only having a sequence, but placing it. It's the first one that was placed in the public domain, and I've never had any negative repercussions from doing that. Had a lot of death threats for creating life, but not from the genome itself.
What did you find most surprising about your genome? And before you answer that, I just see if you remember. So it was after the Scherer genome, Craig and his institute continued to sequence his own genome to very high coverage using Sanger sequencing, and I think it cost probably $100 million in total at the end of the day. And we had collaborated with his group around the copy number variation, and Lars Fuchs was a postdoc, and him and I came down to your institute, and I don't know if you remember, but we walked in the room and presented your C&V data.
Yes.
We were a little bit worried what the response would be, but I'm curious, what did you find most interesting about your own genome?
What I found most interesting is that we couldn't really decipher much of anything.
Right.
And 25 years later, it's not that much better. I think the thing that stuck out most was you and others showing that I was heterozygote for ApoE4. But that led to the formation of human longevity, because I don't know if you want me to go into that story.
Sure,
or not,
please.
but,
Yes.
I decided, because I had this supposed genetic risk for Alzheimer's disease, that I would get a brain MRI and an amyloid PET. But nobody wanted to do it because I didn't have any symptoms, and they didn't feel right doing these tests. Finally, my friend, who was head of neurology, said they would do it under a pseudonym. They're very concerned if they actually believed the genetics, and that if the genetics proved, in fact, that I had the start of Alzheimer's, that it would adversely affect my career. I wouldn't be able to run for president or something, or maybe that was a requirement. So they asked me for a suggestion for a pseudonym, and I said, how about Charles Darwin? And they go, no, that's too obvious. So they made up a name. But they called me a few weeks later, and I think they were very surprised. They said there was no hint at all of any thing to do with any kind of dementia. Amyloid was totally negative. In fact, they said, you have a brain of a 40-year-old. I was 60-something at the time. And my wife said, no, he doesn't. He has a brain of an 18-year-old. But that was the start of setting up to do comprehensive phenotype testing, because it was clear the genetic predictions and what geneticists were coming up with of disease risk from the genome were highly flawed.
Right.
And so what's been lacking in the whole genome field is any genotype phenotype comparisons for the most part, except people like Steve looking at specific genetic phenotypes manifesting as a disease. And so when people looked at people with diseases and found genetic changes, a mistake that many or most made, they didn't go back and look at a broader population to see whether those alleles were truly broadly predictive. I don't know if you remember at Scherer, we did a study. It was 2 hospitals in the UK. We were looking at alleles trying to predict breast cancer therapy success. And we found this one SNP that looked like it was just an amazing correlation. And we'd written a paper, we were about to submit it to Nature. And I decided maybe we should, test on broader populations. And it turned out it totally went away and was a fairly common variant in the broader population. And that just from a lot of inbreeding around these 2 village hospitals in Britain,
Right.
that it led to looking like a unique genetic finding. And I don't know your view of how often that happens in genetics, but we quickly shredded the paper and did not, and we're grateful we didn't submit it.
Yeah, you'll see a lot of head shaking here. This is what we do here. Elise is shaking her head, and absolutely, and it is. I think the new discoveries will come from proper genotype phenotype correlations in properly ascertained populations.
So we've now processed over 8,000 people through the health nucleus at Human Longevity.
Yeah.
And a large number of people hearing about my findings that were ApoE heterozygotes. And almost universally, none of them have any Alzheimer's pre-symptomology. In contrast, though, roughly 85% of the homozygotes do show a tendency. So it's not like it was totally a bogus finding, but probably just assuming if the homozygotes were at risk, heterozygotes must be at half the risk or something.
Right.
But it just vaporized totally. There was no risk prediction whatsoever. And, I don't know your view of BRCA1 and BRCA2, but basically they convey a 50% lifetime risk, not what most people are told. Unless you have a family history of every woman in your family has breast ovarian cancer, then it's like a high 90% chance of risk, which just means BRCA1 and BRCA2 are not the cause of breast ovarian cancer, and the alleles that actually link to the cause are still yet to be discovered. So family history is important, but just those tests on their own are not at all diagnostic. So I'd say the biggest frustration we have is that how little can be determined from the genome itself right now without the phenotype data. But with the phenotype data, we can make N of 1 discoveries all the time. And I can go through one with prostate cancer if there's time and interest.
Okay, we'll come back to that if we have time. So, you've built amazing teams over the years, and I had the opportunity to work with Sam Levy and Mark Adams, Gene Myers, and I think that's one of your greatest strengths. And, I'm now the Chief of Research. I think a lot about team building. Can you comment on the importance of starting your career early? And you did not do a postdoc. You went right into academia is the same for me also. And you want to comment on that?
Well, I did a postdoc in Vietnam before I started.
Okay.
So,
tell people about that quickly.
Well, I got drafted on my surfboard and ended up in the Navy Medical Corps. But in the military, there's no liability rules. So you can do whatever your training allows you to do, and the people you're working with have confidence in you doing. Even though I barely graduated from high school for lots of reasons, none of them have anything to do with academics. I was like a sponge with learning to do medical techniques and information. In age 18, 19, I was training interns and residents how to do spinal taps and liver biopsies. I ran for a year the largest infectious disease ward, at least in the US. And that's where I got introduced to all the diseases I ultimately characterized, meningitis, malaria, tuberculosis, et cetera. And so when I got out of the military, after a year in Vietnam, I was highly motivated to learn, and it was a lot easier. I was 22 when I started college, and I have the shortest PhD on record for the UC system. I published 21 papers in 3 years, and got job offers for faculty positions before I'd even written my thesis. So
I hope our students are listening out there. Did you hear that? 23 papers. So it's important
I work
to start.
at Scripps Oceanographic. I'm on the president's advisory board there for the last 20 years. Graduate students love it so much they take 8 to 10 years to get their degree, which is
that's a problem.
It pays a lot better now to be a student than it used to.
But you think it's really important to start early and get them going? And
well, it depends. I think I benefited from starting later.
okay.
You know, because I was more highly motivated, knew what I wanted to do, and had real world experience in a war. You know,
Right. And this, I think it's in one of your other books, not the Sorcerer, but you talk about adversity in your life and that how shaped you to become a scientist and think about science in a different way and deal with failing, because most experiments fail. I just briefly comment on that.
well, I think optimism and pessimism are genetic traits. I don't know how you can be a scientist and not be an optimist. You know, I mean, it should be a job requirement, or you're spending all your time talking yourself out of doing experiments, so you won't get very far. But you know, I mean, everybody fails. You know, I've been lucky that most of my big ideas have worked even better than I expected, and certainly better than everybody else expected. But you know, I've had some serious problem moments. I started 2 major companies, and I got fired from both of them. After sequencing the human genome and raising $1 billion, the parent company of Scherer, decided I was too much of a liability with my attitudes and decided to fire me. So, that took a while to recover from, but life is about getting on to the next stages, and
right.
It's something optimists do. You just get up, and there's no other choice.
So I have a few comments. So, I mean, you've led all of these massive projects. I hope maybe you'll just tell us what you think if you have a new one coming. But, and you go back and look, you were the scientific visionary and leader in some way. And with the movie Oppenheimer coming out this past summer, there was Oppenheimer, but also General Groves, who kind of ran the operations. And did you always have a Groves behind you in these big projects?
So you might recall at Solera, we had 4 major teams, but you were sort of asking my philosophy to these teams. People sort of start off assuming I'm going to be a micromanager, but I'm just the opposite. I try to hire the best people for the job, mm-hmm. and then turn them loose to do their job. If I was wrong in the judgment, they weren't the best person, that shows up pretty quickly. But we had the team that had to write the new algorithm because with the first genome, we only had 25,000 sequences to assemble. With the human genome, we had 25 million.
Yeah.
So the math, the algorithm, the computation was far more difficult. We had to build a new supercomputer. At the time, a supercomputer was 1.5 teraflops. Some of you probably have desktop computers with that much power. The ABI sequencing machine was a brand new machine. It was the first capillary sequencer. And we had 300 of them, and they had very serious problems. It had a robotic arm that sometimes would go and fly across the room and smash into another sequencer and damage it. So the startup problems were great, but we had a team that was just to manage those. And then Mark Adams was leading the robotic team to produce the 25 million sequence fragments. The library team was the fifth team, but that was basically Hans Smith and one technician. And he did it all himself with his own hands. But each team knew that they had a very precise job to do, and that if they failed, the whole thing failed. So, I couldn't tell the algorithm people how to write a better algorithm.
Right.
And I knew the principles of what I was trying to do. I've never built a computer before, but I knew what we needed.
Right.
And, I liken it to more being an orchestra conductor. The orchestra conductor has to know the score. They have to know something about each of the instruments. But if they could play them, they'd probably be in the orchestra versus conducting it. But,
right. When you look back on your career, what do you see as the best time for you, for your science? What was the most enjoyable time? And I think this may segue into your book, but let's see.
Yeah. So I think it's definitely post the human genome when I had the money, the resources, the team, just to pursue ideas that I was interested in, like making the synthetic cell, like asking questions about the environment and doing things. Those of you who read the book find out it was a fantastic adventure, sailing around the world. And Discovery Channel was filming the part of the trip in the Galapagos, and two of the world's best shark cinematographers were there. I had to learn from them how to dive and swim with some of the largest sharks in the world, which was great training for dealing with geneticists and at NIH and other places. And the trick, if any of you are divers, is by swimming with the sharks as though you're one of them. And there's some, if you can get that, the video from Discovery Channel, I think it's on YouTube and other places. It shows the scene of swimming along with roughly 50 or 100 sharks. And as long as you swam with them, like you were out hunting as well, they left you alone. The most dangerous person was not the cameraman, and these were at the time these very large high definition cameras that they were swimming with. The light guy was the dangerous guy. So if he shined the light on you, that's where the sharks would go. So you wanted to stay behind the light guy. But so a lot of adventures that you wouldn't expect just from doing science. And they came up with this brilliant idea that I should dive down 80 feet of water and grab the dorsal fin of a white-tip reef shark for dramatic effect. And I thought it was a really dumb idea, but it worked out okay. But I was doing another shark dive with a French guide, and his shark belaying tool was a ski pole. And, I had no idea what we're getting into. A little chum bag. We went down roughly 85 feet of water and sat on the bottom, and he opened the chum bag, and you could see roughly 200 feet in every direction. It was like when he opened the bag, it was sort of like you would describe a horror movie of these 8-10 foot sharks just coming out of the blue at you. And because I was sitting on the bottom and I had my fins up, a bull shark thought I looked tasty and kept attacking me. So I got back in that moment, and I kept having to kick it in the face.
Right.
And so I had to develop how was I going to get out of this situation? And on Sorcerer, we had a woman, that position was called a deck stew. And on all the dives, she always sucked down her air faster than anybody else. So I figured when she ran out of air and had to surface and the sharks followed her, I would swim in the other direction. But basically, once all the chum was consumed, all the sharks just dissipated.
Right.
The bull shark that was attacking me was the last one to leave, but he finally gave up. So these kind of stories, plus being shot at in Vietnam, made it easier to deal with all the attacks during the human genome.
And failed grants and reviews.
Yes.
So the book is Voyage of Sorcerer II, the expedition that unlocked the secrets of the ocean's microbiome. And when I was reading it, I kept thinking, we all have these visions of those early days in our labs, those romantic moments where you're pipetting by the glowing refrigerator. Did you have any of those moments here on your sailing ship going around the world?
Well, there's so many unique moments. Just, I had the privilege of two different eras of seeing the Galapagos through Darwin's eyes, and that at the same time with the new lens that we're having. It was a phenomenal juxtaposition of things. And also when we explored the Sea of Cortez, we had John Steinbuch's a book about the Sea of Cortez, and when he went down there sampling marine life with a marine biologist, and so it made the books and the stories come to life, and that you were reliving their experiences, and that made doing the science that much more unique. But you also learn things about geography. Those of you who saw the movie Master and Commander, if you remember, the number one problem is the problem Darwin had as well. There's no fresh water in the Galapagos. So the place everybody goes is Cocos Island off of Costa Rica. It's where the opening scenes for Jurassic Park were filmed, and it really looks like it's in the Jurassic era. I think there's a picture in the book. There's 3 or 4 freshwater waterfalls that fall right into the sea, and the sailing ships would just go in there and refill their barrels of seawater. But there's another picture in there. They would all carve the names and dates of their ships in the rocks around this harbor. So it was seeing history, it was reliving history, while trying to make new history. So it was, there were exciting moments.
Beautifully put. And the book is beautifully written. You have 2 other books. I would recommend all the students read these books. You'll learn so
I
much
recommend
about.
it.
And we actually have we still have some books left.
You can get them from Amazon in 24 hours.
Yeah. So how are we doing for time? Okay, so I have a few. I don't know if you want to make any more comments on the book. Many interesting, more relevant to discovery.
No, why don't we get to, we can take questions from your colleagues.
Okay. So I'm going to add. So for those of you who've seen other Discovery Dialogue interviews, I added a new section here just for Dr. Venter. It's a bit of an experiment. I don't get to do experiments often anymore. So I'm going to just read you some rapid fire questions, and you pick a word, and if you want to add to it later. So DNA, RNA, or protein? What's your favorite?
DNA.
That's pretty predictable.
Depends whether I'm eating it or not, but I prefer protein for diet. So.
Okay, BlackBerry or iPhone?
Oh, iPhone. Well, but BlackBerry doesn't exist anymore, but, or does it up here? I'm sorry. So you might remember it, Lara. So my wife Heather actually introduced me to the very first BlackBerry when it was just a small screen, and we were all everybody at Solera. We bought them all Blackberries, and
so I have to say, so it's a Canadian company. It's from
yes,
Waterloo.
no, I know. I've watched the movie.
Yeah. And the first time I saw someone wearing a holster was actually Craig Venter when I came to visit Solera. So that's why I asked you that question.
Well, back then it was a BlackBerry. I got disappointed when BlackBerry tried to mimic other things versus staying true to.
Yeah.
The keyboard was nice.
Yeah.
You guys are too young to even remember.
Yeah. So how about Watson or Crick?
Oh, definitely Crick. I dealt with both of them. Crick was the true intellectual, and really a nice person. And he was, when I moved to San Diego, he became very fascinated because Watson was so attacking me. And, you know, he's taken on Crick a few times. My institute has this huge scientific archive of papers that were collected by these two scientists, of everybody's scientific papers. So we have Rosalind Franklin papers, everybody. We have her the original Photo 51. But one of the most interesting thing is the original version, draft version of Watson's book, which the original title was Honest Jim.
Right.
And a 6-page carbon single-space typed letter from Maurice Wilkins, line by line, shredding the book and attacking Watson. We have Crick's first and only lab notebook, where he tried briefly to repeat Chargaff's experiments on the base pairing and failed. He couldn't do it. So a lot of interesting things. We, as I explained earlier, we at the Recursive family, we sequenced Crick's genome for them, and they used it for the opening of the Crick Institute. But very, very different people. Watson's ending is much sadder, but it's, you know, his racism and sexism caught up with him. And
okay. People are going to say, what are you talking about? Sherkston Beach, Ontario, or China Beach, Vietnam? If you look right across,
yeah.
you spent time in Buffalo, right across the lake here.
So Sherkston Beach was the hangout for everybody who raised Toby cats. And so when I was in Buffalo, I was a big Hobie cat racer, and took second in the U.S. Northeast Championship. Then I came up for the Canadian Nationals. Nobody ever shot at me on Sherkston Beach. I had trouble with immigration going back and forth all the time on the Peace Bridge, but China Beach was much more dramatic. I used to run 6 miles every day on the beach, and my quote friends in the machine gun towers out of boredom, would shoot at me to see how close they could get without hitting me. And I learned to keep a very steady pace.
Right. I only know
didn't want to trip.
Sharks Beach. My family used to vacation there when we
Is
were
that right?
little kids. I read this, go, oh my gosh. Okay, so now just very quickly, last few. Nature, you published your key papers, Nature, Science, or PLOS Biology. Which journal is the easiest to work with?
PLOS Biology was definitely the easiest and nicest to work with. I'd say most of my key papers were in Science. A few, you might remember, Nature did their first time ever special issue for our giant catalog of all of the human EST sequences. But it got polarized with the human genome papers. Sort of the end of the cooperation, and the journals sort of took on the persona of the different sides. So
yeah. Canadian beaver or American eagle?
Are we talking about dining?
Enough said. Okay, one more, and then just one last question: sailing or flying? You started to fly recently.
Yeah. I started to fly actually when I was in Buffalo, out of Niagara Falls International Airport that got closed down shortly after I did it. But I started again during COVID, and I got my pilot's license at age 72. Flying in the U.S. is sort of the ultimate freedom that's left. I don't have to ask anybody's permission. As long as you avoid restricted airways, you can go wherever you want, whenever you want. And you can't do that in Europe. You can't do that virtually any place else. I don't know about the Canadian rules, but
we have Jason.
It's truly the ultimate freedom where you can just pick up and go. So
yeah.
I fly from San Diego to Maine every summer and back, and I fly in this direction. This one airport, the top of Maine, you have to fly over Canada to come back and land. Right. But after 65,000 miles at sea, I'm not anxious to cross any more oceans.
Right.
But I do have a vintage sailboat built in 1935 and take part in vintage racing on the East Coast. So both are great combinations of using your intellect and using nature and participating in it in a unique way. So,
last question. If you were a new PI starting at, say, SickKids, for example, is your first job, and you could hire one new person to start your lab and your career, who would that be? Either name or phenotype or whatever you want to share.
Well, the best colleague I've had in my career is Ham Smith. He and I met at a meeting in Bilbao, Spain, on the ethics of genomics. This was before everything started, and I was talking about ESTs, and the Catholic priest got up at this meeting and started attacking me because NIH was patenting the EST sequences, and he said, you shouldn't patent life. I said, all life, or is it just human life? He said, just human life. And I said, well, how do you reconcile when some of our human genes have the same exact sequences as a pig gene or mammal genes? And that did not compute, and he just sort of went away frustrated. But Ham got up to ask a question, and the first thing he said, he goes, "Where are your horns?" Because I was supposed to be the devil, and then we had drinks that night at the bar and became fast friends. He was going to retire from science. He got his Nobel Prize in 1978, and he was frustrated. He was in his early 60s, He's frustrated with the grant system and the Me Tooism in science, and he was going to retire. And then he decided to come first to advise my group and decided to join it, and had a second career that I think was even better and more productive than his first. He's been my best friend in science, a great constant mentor and colleague and friend. And, you know, if people can achieve that kind of relationships in science, they'll have a very good life.
What a great answer. So the take-home message is, if you're starting your own lab, try to recruit a Nobel Prize winner to work with.
They have a lot of influence.
You know, they may be looking for a postdoc.
Yeah. I described Ham Smith and Clyde Hutchinson, who retired from North Carolina, as the two best postdocs I ever had. And it turns out this is a fairly common phenomenon of people that sort of the later end of their careers go back to the freedom that some of you have as postdocs, to just go do research in an unencumbered fashion.
Yeah.
And so they're equivalent to postdocs in their own minds as well. And it's a great opportunity.
So any last words before we open up for questions? And before we do that, I just wanted to thank you. You've been very generous with your time, not only on this trip, but over your whole career for SickKids and for Toronto and Canada as a whole. It's been a great day, but any last words?
I've enjoyed it thoroughly. Please don't give me a career development award yet, because I'm not done yet. I've started to get several lifetime achievement awards. I said, can you wait a while? I still got more to go. No, so.
Okay, great. Thank you so much, Craig. Are there any questions from the audience? We have lots of students and postdocs and some PIs here.
I can't hear very well.
Being a scientist, what is the best quality in your opinion?
Quality of being a scientist.
The best quality. The single most important trait is high integrity. And followed by curiosity and willingness to work hard. It's a good combination, but
that's the first time I've heard integrity. You usually hear stubbornness, and but you're right. You're right.
25% of papers published now have fake data in them, and there's 20 times more scientists alive today than when my career started. And our early mentors were able to pretty much control the field of science. And when people stepped out of bounds, they were able to intervene, either blocking their publications, blocking their future grants. And now it's gotten too big that nobody can really play that role except on a local basis. But just think, with all the things that I've done in my career, and most especially if I'd left the slightest hole open in the integrity of our work, the integrity of our approaches, Francis Collins and other people would have stomped through them, and the data's less, the period of time, and that's, there's bioethicists, but they're not the preachers of science. They're not the priest of science. Every one of you has to be your own bioethicist, and the decisions you make every day, just like the decisions that we make, are what determines the future of science. So that's why I put it as number one. It's probably more important than it's ever been, and with ChatGPT producing even more crap, it's going to be even more important.
Yes, just stand up and maybe project. Yes.
Just on the back of that, with ChatGPT, with the development of AIs and generative AI, how do you think that could accelerate discovery? For example, on projects that you run yourself, like minimal genome project, how could that help that develop that further?
So could everybody hear the question? Okay. So this comes up a lot. AI has been around for over 50 years in various forms and various algorithms. What I argue is AI is only as good as the training sets that it's trained on. And the trouble with ChatGPT is trained on everything that's available on the internet, which is a lot of crap, as well as a lot of good things. And so it's, it's like having your memory being half decided by Donald Trump and half by Barack Obama. And so with that as a training set, you don't want to trust anything that comes out of it. But for science, one thing that's been good, it's improved the 3D predictions of proteins, which has helped us unravel some of the unknowns in the minimal cell. Those algorithms still have a ways to go, according to some of my colleagues, but it was an improvement. I think the biggest challenge for people, it already is a challenge on a daily basis, sorting out fact from fiction. And, but it can't solve unknowns. It can't just say, what do these genes do without a reference. So that the training sets become more and more important in science. And if you're trying to ask a question, you need to make sure you have the right training set, or it will just mislead you.
Questions at the back? Just stand up and project, please.
So you answered a lot of questions throughout your career. And I guess what I'm wondering is, what are the questions you are still asking yourself today? What do you continue to be curious
about? I don't think we've succeeded in answering most of the questions that I set out. We've taken the definition of life a long way with the minimal cell, and we've created a tool where you can now recapitulate evolution by adding genes back to it. I would have hoped by now we could have understood human life far better from the genome. It's the same frustration I had first looking at the Haemophilus genome, and was just sadly disappointed that we couldn't describe the life of the cell from just looking at genetic code, because we still don't have the knowledge basis to do that. So we can't do that with a minimal cell. We're still at roughly the 1% level for the human genome. I know there's life on other planets. I'd like to see it proven in my lifetime, but I do already know the answer, so it doesn't really have to be. But I described this in my second book, Life at the Speed of Light, about making synthetic cells and making the first biological teleporter just by sending a sequence through the internet, through radio waves. We can send it long distances and recapitulate life at the other end. And we've done this with viruses, and we exchange about 100 kilograms of material every year with Mars. So you can't take a scoop of Earth here without having Martian soil in it. So when microbes or their remnants are discovered in the subsurface oceans, they'll resemble life on this planet, because there's been the exchange of material already. So I think it better be a great expansion of human consciousness to have it proven that once again we're a minor artifact in the universe versus being the center of everything. But I haven't lost my curiosity.
Nicely put. Last question.
We can't
You
hear
have
you.
to speak louder.
Okay. I thought you were asking me what I do in my private life versus my public life, but um, it was very simple. The U.S. government was not going to give me money to sequence the human genome. Collins tried to block us getting money for the first genome. He didn't like the notion. They wanted to have their multi-billion-dollar public works project. Somebody offered me $300 million to start a company to sequence the human genome. You know, the only reason I took it is I was certain that I could do it. Otherwise, it would have been one of the biggest burnouts in scientific history. But, you know, the notion that one place is evil and the other place is good is a very, very old view of the world. Some of the best science comes out of private industry, and some of the best crap comes out of academia, and vice versa. So more and more people are leaving their PhDs and postdocs and going into industry because it pays better and you don't have to compete for grants. But that's also a lazy way out, because then you're working on other people's ideas instead of your own, unless your ideas align with the goal of the enterprise you're joining. But there's no panacea. I've worked with some of the best people in both worlds, and some of the nastiest people we've encountered are people that hide behind academic screens. So the world's made up of humans, and some have integrity and some don't.
Okay, one very last question. Please stand up and come closer to the microphone.
I'm just really curious, how did you manage to publish 21 papers in 3 years?
Get back to work.
Like what kind of hard work did you put in through this process? Like did you just do it nonstop, without any sleep?
And they were all in Science and PNAS and top journals too. Well, I was very, very motivated. I was doing unique experiments. I was doing them all myself, and I was young enough that I need less sleep than I need today, and you know, it was a pretty exciting phase and opportunity that I could do that. Right, it helped. My mentor was a member of the National Academy. The old rules before I got in. Mentors could submit 8 papers a year, and they were automatically accepted. So that, you know, that helped quite a bit. But my work was very controversial from the beginning. I proved that the adrenaline receptor was on the outside of the cell by attaching adrenaline covalently to glass beads and showing that it still worked. And so it created a lot of excitement and controversy at the same time. But Steve said it earlier today, being a good writer is part of being a good scientist, because scientific communication is essential. And also, writing 3 books took a lot of discipline. It was hard to do. I wrote a lot of the first one while sailing around the world. My agent actually talked me into writing the books myself. You know, I studied some writers who gave advice. Hemingway probably has the best advice for writers. He said, if you're going to write, you have to write every day. And same discipline I set for writing scientific papers. I got up every morning and spent an hour writing. And some days I'd write a word or a sentence, and some days a few thousand words. And it was such an enjoyable discipline that I almost hated finishing books because I enjoyed the process so much. I wrote most of my papers and my thesis between roughly 1 a.m. and 4 or 5 a.m. because it was the most peaceful time and I could have it to myself without interruptions. But, you know, it's setting up the discipline to do it. You know, everything, success in any field that I know of, there's a certain amount of luck, but the rest is hard work and discipline.
10,000 hour rule. You know, the best advice I got was from my PhD supervisor, and it was to read a paper in your area you're doing the research, read a general paper, and read a technology paper. So 3 papers a day, and that's how you learn how to write. You'll mimic what you're reading. So read, and write. Practice your writing.
I read less. I was too busy writing.
Doing experiments. Okay. Thank you again, Dr. Venter.
Thank
This
you,
has
guys.
just been wonderful.
Yeah. Fun conversation.